Hi everyone, my name is Juana Cecilia Martin Gonzalez. I'm currently a student here at the University of California, Riverside. I work under Dr. Sandi's laboratory in understanding the formation of spherical viral capsids using statistical mechanics. So what is a virus? A virus is basically the simplest physical object in biology. It is an infectious agent. It is composed of RNA or DNA. It contains a coded protein called the capsid. And in terms of its size, it is in nanometers. In comparison to something that we're familiar with, such as a human hair, it is about 100 times larger. Like a Zika virus is 0.045 micrometers, and a human hair is 60 to 120 micrometers in diameter. So why study viruses as a theoretical physicist? One of the main reasons is because laboratory experiments are simply not enough. When it comes to laboratory experiments, we can only see proteins, free subunits, or fully formed capsids, but nothing in between. So we don't actually know how this virus is fully formed in the process or the intermediate steps. So one of the great ways that we can learn about this intermediate step is using the theory in physics to provide greater insight of viral capsid formation and use that for treatment and therapy. So viruses and physics. To study the assembly of empty spherical viral capsids, we use something called classical nucleation theory, which takes into account how a single protein is being added at a time, forming these clusters, and eventually this partial capsid of size n. And if it overcomes an energy barrier, which originates at the rim of the partial capsid, due to the missing protein-protein interactions, it forms into a fully formed capsid. And this video kind of demonstrates how a single protein is just being added at a time, forming a fully sized capsid. And one of the ways that we actually use to experiment on classical nucleation theory is using viruses such as the copy-chlorotic model virus, also short for CCMV, which is an infectious copy plant. It is spherical. It is very easy to remove the RNA or DNA, and we can simulate the self-assembly of the protein subunits into shells of identical sizes to the virus. So I know that I said that viruses are spherical, but in terms of their capsid structure, they're actually icosahedral, which is very close. It has 20 triangular facets, and in terms of their size, it depends by triangulation number, which is the number of proteins in each face. In this case, we see how a T1 is 60 times 1, so it's 60 subunits that make up this T1 capsid. And for a T3 is 60 times 3, so it'll be 180 proteins, making up a T equals 3. So how do we actually test the classical nucleation theory? And one of the ways that it was experimentally tested was using the copy-chlorotic model virus. They added or they attached elastic-like polypeptides at the end of the protein or the N-terminal, which made it hydrophobic. So what happened is we had a T equals 3 capsid, and as it disassembled by increasing the pH from a 5 to 7.5, these proteins went into free subunits in solution. They become hydrophobic, meaning they didn't want to be surrounded by water, and it forced it to form a T equals 1 capsid. In a similar way, if we decrease the pH from a 7.5 to a 5, these proteins disassembled and assembled into a T equals 3. And this were the results from the experiment. What we see here is in this diagram, we see how a T equals 3 capsid is disassembled and to proteins free in solution in the yellow, and a T equals 1 is formed as the pH goes from 5 to 7.5. The data collected is actually the squares and the dots, and the solid lines is classical nucleation theory. This actually verified the classical nucleation theory did an amazing job in actually predicting both the assembly and disassembly for both the T equals 3 and T equals 1. And very similarly, when we lowered the pH from a 7.5 to a 5, we see how a T equals 1 in blue starts to disassemble, and in yellow, it starts to assemble that T equals 3. And one of the equations that I actually got to work with was the master equation. And as complicated as it seems, is not necessarily as complicated. What we see here, it actually explains what we can't see in the laboratory. This is how a partially formed capsid of size N is formed by either losing or gaining a protein. So if it gains a protein, it takes into account the rate of attachment, and if it loses the protein, is the unbinding rate. So partially formed capsid of size N can form either by gaining a protein from an N-1 partially formed capsid or losing a protein from an N+1 partially formed capsid. And in a very similar way, we can also study the formation of a partial capsid of size N-1 and N+1, where C is the concentration, N is the number of subunits in a partially formed capsid. And finally, we have the steady rate of capsid formation. And this equation explains why is it that we cannot actually see in laboratory a partially formed capsid. And the reason why is because it's mathematically an exponential. That means it very quickly either forms into fully formed capsid or it goes back into free subunits. So this is amazing because it takes into account in delta G star the activation energy, which is the energy barrier that again, it's associated with those proteins or the missing protein interactions at the rim of this partially formed capsid. The Seldovich factor is a small correction that takes into account how this partially formed capsid can recross the energy barrier and go back into free subunits in solution. And the beta star is the rate of attachment to the critical nucleus. So how quickly these proteins are being attached to the fully formed capsid. And CS is the concentration of reference state. And in terms of the future research, it includes the polymorphism of viruses, nanotechnology, and applied theory to more complex viruses. And I just want to thank Dr. Sandy, graduate student Ali Reza, my group members, Jennifer and Jessica, that I got to work with, and the California Alliance for Minority Participants, alongside with the Lewis and Stroke Alliance for Minority Participations. And here are my references. Thank you so much. If you have any questions, let me know.
That was a great talk, Juana. Thank you very much. Do we have any questions from either the chat or anyone that would like to question